A camera descended through almost 7 miles of black water and stopped just above the floor of the Mariana Trench. Its lights opened a small white circle on the sediment. A piece of fish had been fixed to the lander as bait. For a while, the frame showed almost nothing, until small pale bodies began moving through the light. They were not fish, but shrimp-like amphipods. They were amphipods, shrimp-like crustaceans built for a part of Earth where the pressure is roughly 1,000 times greater than at sea level. The lander was operating in the Challenger Deep at about 10,929 meters, close to 36,000 feet below the surface. That is near the deepest measured seafloor in the world. This is the real footage behind a claim that has been repeatedly inflated online. A mysterious creature was not newly filmed at the bottom of the Mariana Trench, in defiance of every law of biology. Scientists have known for decades that animals live there. Japanese vehicles recorded deep-dwelling crustaceans and worms in the 1990s. Later landers brought back amphipods from almost 11 kilometers down and recorded them gathering around bait. The discovery is still extraordinary, but for a more precise reason. The animal in those images was living nearly 2 miles deeper than the deepest confirmed fish. At that depth, the environmental problem is not simply darkness or cold. Water pressure changes the behavior of proteins, membranes, enzymes, and every chemical reaction that keeps a body alive. A vertebrate solution that works at 6 or 7 kilometers appears to reach a limit before the ocean floor does. Amphipods found another route. The history of reaching that depth explains why the evidence is so hard-won. A vehicle that descends to Challenger Deep must survive pressure that can implode ordinary housings, carry its own light into total darkness, know its depth accurately, and still return data or samples after hours without direct human access. Keiko's 1995 dive reached 10,911.4 meters and recorded several kinds of organisms, and Nereus returned in 2009. Camera landers later waited on the bottom without a pilot, using timed releases and buoyant spheres to climb back to the surface. Every successful return removes another possible source of error from the evidence. A pressure reading rules out footage from a shallower slope. A recovered specimen rules out a reflection or drifting fragment. Repeated encounters rule out a single accidental visitor carried down with equipment. The animal's genes and chemistry then show that it is not merely enduring a brief descent. It belongs to an adapted population that lives and reproduces in the Hadal environment. That difference creates the central mystery of the entire expedition's story. How can a small crustacean remain active near 36,000 feet when fish disappear thousands of feet above it? And why does the bottom of the trench, which looks like a barren plain under artificial light, support animals that can rush toward a meal, digest it, reproduce, and maintain their cells under pressure that can destroy an unprotected machine? First, the depth itself needs to be stated carefully and honestly. 36,000 feet is a rounded description, not a single universal number for the entire Mariana Trench. The trench is a long subduction feature with slopes, basins, ridges, and several deep pockets. Challenger Deep contains the greatest reliably measured depth in the global ocean. A modern analysis of submersible transects estimated its deepest seafloor at 10,935 meters, with an uncertainty of 6 meters. Converted to feet, that is just under 35,900 feet. A 2014 Schmidt Ocean Institute expedition deployed a camera-equipped vehicle called the LEGO Lander in the eastern part of Challenger Deep. It reached a measured depth of 10,929 meters. The baited system recorded the scene and collected 2 kinds of amphipods, including Hirondellea gigas. Another small amphipod from that expedition, a species of Hallis, was later analyzed after being collected from 10,908 meters. Those figures matter because they establish 3 separate facts. The camera system reached the near-bottom environment, animals approached the lander there, and physical specimens were recovered from measured depths close to the ocean's maximum. The story does not depend on a blurry shape crossing an unlabeled video. It rests on depth records, recovered animals, laboratory work, and multiple expeditions. Hirondellea gigas does not look like a movie monster. It is a pale, laterally compressed crustacean, usually only a few centimeters long. Its body resembles a large sand hopper more than an alien predator. Yet it is one of the dominant mobile animals at Challenger Deep. Researchers have captured it at depths around 10,897 to 10,929 meters, where hydrostatic pressure approaches 110 megapascals, or roughly 1,100 atmospheres. At sea level, the atmosphere pushes on every surface with a pressure we barely notice because our bodies are balanced against it. Descend through the ocean, and the weight of water adds another atmosphere about every 10 meters. At nearly 11 kilometers, the pressure is not a sudden crushing blow coming from one direction. It acts from every direction and reaches into the molecular machinery of the body. A gas-filled cavity becomes a major liability, and although flexible tissues help, softness alone is not enough. Pressure can force proteins into shapes that no longer work, alter the fluidity of cell membranes, disturb the binding of molecules, and slow or redirect chemical reactions. The animal cannot simply be strong. It has to be chemically organized for this environment at every level. That is why the phrase "shouldn't exist" is powerful but scientifically wrong. Nothing visible in the footage violates physics or known biological principles. The amphipod survives because its biology obeys the conditions more completely than ours does. It carries pressure-tolerant proteins, protective small molecules, unusual digestive tools, and a body whose structure has been shaped by life in the Hadal zone. The frightening image is an impossible creature beneath a mountain of water. The more interesting reality is a creature for which that mountain has become normal. And the next part is stranger, because the deepest animal is not only resisting pressure, it is solving another problem that should be just as severe: how to find enough food at the bottom of a trench where sunlight has never reached and plants cannot grow. Nearly all energy entering the deep ocean begins far above, in sunlit water. Tiny organisms grow, animals feed, and organic particles sink. Most of that material is consumed or decomposed long before it reaches the Hadal zone. By the time a fragment falls 10 or 11 kilometers, it has passed through an enormous column of hungry water. That combination should make Challenger Deep an ecological desert. It is cold, permanently dark, isolated by steep walls, and distant from the surface production that powers most marine food webs. Yet baited cameras can still be surrounded by amphipods. The reason is partly that a trench is not merely a hole. Its shape can funnel sediment and organic debris downslope, concentrating material in the deepest basins. The trench's shape may also create short-lived abundance from long periods of scarcity. Earthquakes, landslides, currents, and sinking particles can move organic matter downslope. Material reaching a broad abyssal plain can spread across a huge area, but material entering a steep trench may be channeled toward a narrower basin. A bait package imitates a concentrated fall of food and reveals how quickly scavengers can exploit it. This helps explain why footage can look crowded even though average food supply is low. The camera records a biological response to a pulse, not a permanent swarm occupying every square meter. The distinction matters because it prevents another exaggeration. Abundance at bait does not mean the deepest seafloor supports the same biomass or diversity as shallower water. Hirondelia gigas is built to exploit rare opportunities. When a carcass or fragment of plant material arrives, the animal cannot afford to treat it casually. Studies of its digestive enzymes found an unusual ability to break down plant-derived carbohydrates, including cellulose. Researchers lowered baited traps in an 11,000-meter camera system to 10,897 meters in 2009 and recovered 185 amphipods. Every captured animal was Hirondelia gigas. Laboratory analysis identified enzymes capable of attacking cellulose, xylin, mannan, starch, and related compounds. One purified cellulase converted cellulose into sugars and remained effective under high pressure and low temperature, resembling conditions in Challenger Deep. The results suggested that the amphipod can use wood and other plant debris that has fallen from the surface and become buried in trench sediment. That does not mean the animal lives on driftwood alone. Amphipods are opportunistic scavengers, and baited landers attract them because a piece of fish is an unusually rich energy source. The broader point reaches beyond one unusual digestive enzyme. In an environment where meals are irregular, survival favors an animal that can digest several kinds of material and consume a large amount when food appears. Its chemical defenses continue outside the gut. Deep-sea organisms accumulate organic molecules called osmolytes that help proteins keep their shape under pressure. In amphipods, the balance of those molecules changes with depth. Compounds associated with shallow water decline, while pressure-counteracting molecules such as trimethylamine N-oxide and scilo-inositol increase. These molecules do not create anything resembling an invisible force field. They alter the chemical environment around proteins and membranes, reducing some of the destabilizing effects of pressure. The solution is costly and complicated, but it allows biochemical reactions to continue where ordinary versions of the same proteins would fail. Genetic studies of Hirundella gigaesis add another layer. Amphipods collected at 10,929 meters showed changes and expansions in gene families connected to energy metabolism, genetic information processing, cold response, and protein function. Scientists do not interpret one gene as a magic deep-sea gene. Adaptation is distributed across many systems because pressure challenges the whole organism. Then researchers found something even more unexpected on the animal's exterior. The exoskeleton of Hirundella gigaesis contains aluminum on its surface. Aluminum is scarce in seawater but abundant in seafloor sediment. Under trench conditions, the amphipod appears able to produce compounds that extract aluminum ions from the mud. In seawater, those ions form an aluminum hydroxide gel that coats the body. Experiments indicated that this coating can protect calcium carbonate in the exoskeleton from pressure-related dissolution. The amphipod is not wearing a metal shell in the ordinary sense. It is using chemistry to build a thin protective layer from material in the trench itself. That adaptation gives the creature that shouldn't exist story a better physical image. A small scavenger moves across the deepest seafloor carrying a pressure-resistant chemical coating assembled from the mud beneath it. Inside the body, osmolytes protect proteins while digestive enzymes unlock energy from debris. Its membranes, metabolism, and genetic machinery are tuned to cold, scarcity, and enormous pressure. None of these systems guarantees survival by itself. Together, they move the boundary of animal life almost to the deepest point of the ocean. The footage also corrects a common misunderstanding about pressure. People often imagine a deep-sea animal as a delicate object somehow holding back the entire ocean. But an amphipod contains water-rich tissues with little compressible gas. The pressure inside and outside the body remains largely balanced. The challenge is not that the animal must support the water like a roof. The challenge is that its molecules must continue working while immersed in that pressure. This is why bringing hadal animals to the surface can damage them even when the trip upward removes pressure rather than adding it. Their proteins, tissues, and membranes are adapted to a narrow physical world. Rapid decompression, warming, and changes in dissolved gases can destroy the conditions on which their bodies depend. A specimen on a laboratory tray is not simply the same creature in a safer place. Modern pressure-retaining samplers are designed to recover amphipods while preserving more of their in-situ pressure. In 2022, researchers reported a sampler that returned 177 Mariana Trench amphipods while retaining pressures above 93 megapascals. That technology matters because many biological processes can change before a conventional sample reaches the ship. A video shows behavior, a trap supplies anatomy and genetics, and a pressure-retaining chamber preserves physiology. Each method answers a different question, and together they reveal an animal that is not merely present at depth, but actively functioning there. The strongest evidence is therefore not a single dramatic frame. It is the convergence of camera footage, measured depth, recovered specimens, enzymes that work under pressure, protective chemistry on the exoskeleton, and genetic patterns consistent with hadal adaptation. But one major question remains unresolved at this point. If evolution can carry an amphipod almost 11 kilometers down, why does it appear to stop fish much higher on the trench wall? Fish dominate almost every familiar marine environment. They live beneath polar ice, around hydrothermal vents, in oxygen-poor basins, and through a vast range of temperatures and salinities. The Mariana snailfish has pushed the vertebrate body deeper than nearly any other fish. It is translucent, soft, scaleless, and so lightly ossified that parts of its skull remain incomplete. Those features are not signs of biological weakness or failure. Heavy mineralized structures are difficult to maintain under hadal conditions, and a flexible body reduces some mechanical problems. The fish also carries molecular adaptations that stabilize proteins, maintain membrane function, and support transport through cells at high pressure. In the Mariana Trench, snailfish have been filmed to about 8,178 meters. A broader review identified the Mariana species Pseudolaparis swirae as one of the deepest known fishes, with confirmed collection and video records around 8 kilometers. In 2023, researchers announced a new bony fish depth record from the Izu Ogasawara Trench, where a snailfish was filmed at 8,336 meters. That record is spectacular, but it remains far above Challenger Deep. The difference between 8,336 and 10,929 meters is about 2.6 kilometers, more than 8,500 vertical feet. At the bottom, cameras can record amphipods and other invertebrates, but no verified fish. The fish boundary is also an example of why pressure resistance cannot be reduced to body softness. Mariana snailfish have flexible skeletons and gelatinous tissues, but their deepest range is set by chemistry as well as anatomy. Their cells rely on TMAO to keep proteins stable, and the amount required rises as pressure increases. Eventually, the protective strategy collides with the animal's need to regulate water and salts. Amphipods use some of the same protective molecules, but their physiology is not organized like that of a bony fish. They can combine osmolytes with different membranes, enzymes, exoskeletal chemistry, and life history traits. Evolution did not give one group a stronger version of the same design. It produced fundamentally different solutions with very different depth ceilings. The likely explanation involves the same pressure-protecting molecule used by many deep organisms, trimethylamine N-oxide or TMAO. In bony fish, TMAO concentration generally rises with depth. It helps stabilize proteins against pressure, but it also raises the concentration of dissolved material inside the body. Marine fish must constantly regulate the movement of water and dissolved salts. Their internal fluids are normally less concentrated than seawater, so water tends to leave their bodies. As TMAO levels rise to counteract greater pressure, the internal fluid approaches the concentration of surrounding seawater. Researchers propose that at roughly 8,000 to 8,500 meters, the fish would reach an osmotic boundary. Going deeper would require a major reversal of the normal water balance system. This is not a solid depth wall stamped across every trench. Temperature changes pressure tolerance, species differ, and the exact lower limit has shifted as better cameras reach new locations. The 8,336-meter record slightly exceeded an earlier simplified estimate, yet it remained within the broader predicted band of roughly 8,200 to 8,400 meters. The key fact has survived repeated testing. No confirmed fish inhabits the deepest quarter of the ocean, from roughly 8,400 meters to 11 kilometers. Camera surveys across multiple trenches reveal a consistent transition. Fish disappear while invertebrates such as amphipods continue downward. That is the honest meaning behind the corrected title. The creature near 36,000 feet does not break the limit of life. It lives beyond the known limit of fish because an amphipod is solving pressure with a different body plan and a different biochemical toolkit. The comparison also prevents a common piece of visual misinformation. Many viral Mariana Trench videos combine footage from different depths and locations. A glowing jellyfish filmed several kilometers down, a Dumbo octopus from another trench, a Mariana snailfish at about 8 kilometers, and amphipods from Challenger Deep may be edited together as though one camera encountered them all on the deepest seafloor. Accurate depth labels therefore matter far more than dramatic editing. A creature filmed at 4,800 meters is living in an extreme environment, but it is not at 36,000 feet. A snailfish filmed at 8,178 meters is a record-setting vertebrate, but it is still more than 2,700 meters above the deepest amphipod collection sites. The same trench name does not make every image a Challenger Deep image. Even within genuine Mariana footage, a baited lander changes what the camera sees. The bait releases odor into the water and creates a concentrated food signal. Mobile scavengers arrive from well beyond the camera's narrow pool of light. A bare patch of sediment that looked empty can become crowded after enough time. The animals were already part of the ecosystem, but the lander reorganizes their behavior around one artificial meal. That is why James Cameron's 2012 view from the bottom and the later baited camera sequence can look completely different without contradicting each other. Cameron described a desolate, nearly featureless plain during his short exploration. A stationary lander carrying bait can wait for hours and draw animals into a fixed field of view. The technology determines which part of this hidden reality becomes visible. A crude submersible can move, observe geology, and place a human at the bottom, but dive time is limited. A remotely operated vehicle can manipulate samples but requires an extraordinary cable or autonomous design at full ocean depth. A free-falling lander can remain quietly on the seafloor, record for long periods, and return with traps and sensors. In 2009, the hybrid vehicle Nereus reached Challenger Deep, collected sediment and biological material, and returned images from nearly 11 kilometers down. Earlier, Japan's Kaiko had reached 10,911 meters and recorded organisms including crustaceans and polychaete worms. These missions established that visible animal life at extreme depth was not a single expedition's mistake. A 2014 camera lander then tied the behavior to specimens that could be examined in detail. The lineage of evidence runs from seeing movement, to recovering the animal, to sequencing genes, testing enzymes, examining the exoskeleton, and designing equipment that keeps the sample under pressure. The mystery therefore becomes sharper rather than weaker. The bottom of the trench is not filled with monsters. It is occupied by a small number of highly specialized forms that have crossed filters other animals cannot cross. Fish approach the boundary and disappear, while amphipods continue into the final kilometers. The camera therefore reveals no violation of nature. It reveals where one evolutionary solution ends and another takes over. The Mariana Trench often appears in popular culture as an empty crack waiting to hide something enormous. The real ecosystem is almost the opposite of that popular image. As depth increases, the variety of large animals generally declines. A few groups become dominant because only a narrow set of biological designs can function under the combined pressure, cold, darkness, and scarcity. At Challenger Deep, amphipods can become abundant around food, while sea cucumbers and giant single-celled organisms called xenophyophores have also been documented in deep trench expedition imagery and samples. Microbial life remains active in both sediment and water. The deepest ecosystem is not lifeless, but neither is it an unrestricted refuge where any bizarre animal could be hiding unchanged. Every resident must pay for depth through adaptation. Cell membranes must remain fluid enough to operate. Proteins must fold and bind correctly, and genetic material must still be copied and repaired. Metabolism must deliver energy from irregular food, structural materials must resist pressure-related chemistry, and reproduction has to work without sunlight. plants, or easy movement between habitats. Hirondellia gigas succeeds because the entire organism is organized around those constraints. Its success is not proof that pressure does not matter. It is direct evidence of how severe the environmental filter has become. A shallow water relative dropped to Challenger Deep would not become Hadal simply because its body contained no air pocket. Its enzymes and membranes would be pushed outside their functional range. This also explains why the animal's ordinary appearance is scientifically valuable. Extreme environments do not always produce visually spectacular or monstrous shapes. Some of the most important adaptations are invisible. A substituted amino acid, a higher concentration of an osmolyte, a membrane rich in different fats, an enzyme that remains active at 100 megapascals, or a gel coating only micrometers thick. The camera gives those hidden systems a moving body. An amphipod enters the light, locates the bait, bends its jointed limbs, and feeds. Each ordinary movement demonstrates that thousands of pressure-sensitive reactions are succeeding at once. The word footage can also create false confidence because deep-sea images are easy to detach from their metadata. Color is produced by artificial lights, and distance is difficult to judge without a reliable scale. A small animal close to the lens can look enormous. Sediment disturbed by a thruster can resemble smoke, and compression artifacts can turn a transparent limb into a sharp edge. This is why the most reliable deep ocean stories are built from unglamorous documentation. Coordinates, pressure calibrations, recovery notes, specimen labels, and archived video make it possible for later researchers to test the original interpretation. Without that chain, a striking frame may be useful for curiosity but weak as evidence. The phrase, captured a creature, needs one final distinction. Camera footage captured the animal's behavior, while traps captured physical specimens. Both happened during Hadal expeditions, but they are not interchangeable. Video alone may not identify a species with certainty, whereas a specimen allows anatomy and DNA analysis, and a depth sensor establishes where the encounter occurred. Strong claims require all three lines of evidence to align. For the 2014 LEGO Lander expedition, the camera payload, measured deployment depth, and recovered amphipods provide that alignment. For Kaiko, the dive record documents video of organisms at more than 10,900 meters and later collection of Hirondellia gigas. For the deepest fish records, baited camera deployments and depth instrumentation separate verified observations from old reports whose locations could not be confirmed. This is why the final title cannot honestly say the animal should not exist. Science did not predict a sterile Challenger Deep and then watch an impossible being appear. Deep trench sampling had already shown that invertebrates could reach full ocean depth. The surprise lies in the mechanisms and in how far the limits differ among animal groups. A better statement is that the footage captured a creature near 36,000 feet, beyond the known fish limit. That contrast is supported by what cameras and specimens have repeatedly shown. It also opens the real question, not whether life can exist, but which forms of life can cross each physiological boundary. The latest research continues to refine that biological map of the trench. A 2026 study used computer vision to reconstruct the three-dimensional swimming behavior of Mariana snailfish from lander video recorded at 6,670 meters. The work did not move the fish depth record to the bottom. Instead, it showed how slowly and economically a Hadal vertebrate searches for food in its natural environment. That is a useful current update because it connects behavior to the depth ladder. At around 6 to 8 kilometers, snailfish remain active predators of amphipods. They are soft-bodied specialists rather than helpless animals drifting with the current. Farther down, the vertebrates vanish, but their prey continues into Challenger Deep. Technology is also narrowing the gap between an animal in the trench and an animal in the laboratory. Pressure-retaining samplers, full ocean depth submersibles, autonomous landers, environmental DNA, and improved cameras allow researchers to ask questions that older expeditions could not. Scientists can compare behavior, chemistry, genetics, and physiology without assuming that a decompressed specimen tells the whole story. Major unknowns still remain. Researchers do not know every pathway that protects Hirondellia gigas, how populations move among separate trenches, how often food reaches the deepest basins, or how trench circulation distributes oxygen and nutrients. A camera sees only a small area. Bait favors scavengers, and traps miss animals that avoid the odor, so every method carries a measurable bias. Those limits should make the story more compelling, not less. The deepest seafloor is one of the few places on Earth where a basic inventory of visible animals remains incomplete. New species remain possible, and additional adaptations are expected to emerge from future work. But a responsible documentary must not turn that uncertainty into evidence for a fabricated monster. The animal already visible in the verified footage is extraordinary enough. Picture the lander resting on fine sediment beneath almost 11 kilometers of water. The light reaches only a few meters before darkness absorbs it. Pressure surrounds the camera, the bait, and every moving body at roughly 1,100 times the pressure of the atmosphere. A pale amphipod slowly crosses the illuminated edge of the camera frame. It does not know that the depth is a record, that fish ended far above, or that its enzymes and exoskeleton have become evidence in laboratories across the world. It follows a chemical trace toward food, because, in the environment that would destroy most familiar animals, its body is operating exactly where evolution placed it. That is not a creature that should be biologically impossible. It is a creature that shows how much life can change before existence becomes possible.